Good evening. My name is Pani Descossard. Today I'm presenting a new topic for the channel Quantum Origins. The topic is eukaryogenesis, or the origin of the eukaryotic cells, the cells that make plants and animals, fungi, humans like you and me. And in this context, we'll present a new theory to explain this major event in the history of life, the bilateral descent theory of eukaryogenesis. This is a theory that has been exposed and explained in the book Ways of Nature that we published in November of 2023. So the major elements of this theory is exposed are exposed in this book. And if we ask any student or academics or scientists, biology or physics, about this major event of the history of life, eukaryogenesis, the appearance of eukaryotic cells, and everybody will go to the endosymbiotic theory that has been developed by the late Professor Lynn Margulis. What I've said in this at the beginning of this channel is that quantum origins, the channel quantum origin is there mostly to challenge previous theories. We're going to challenge and propose new theories for the study of science, particularly biology and physics. Um, Here, a timeline of the history of life. On the left, 4.5 billion years ago. This is when our planet Earth was formed, and within about a billion years, it's about 2.5 billion years ago, 2.8 billion years ago, we have evidence of prokaryotic cells, such as we have some fossils of what appear to be bacteria, for instance, in that found in the rocks, in some rock samples in Australia. We have the stromatolites, which are physical structures formed by cyanobacteria, again in Australia. And for more than 2 billion years after that, according to the fossil record, um, the prokaryotes, bacteria, and archaea, so seems to have been the only cell-based living organisms. There were no eukaryotic cells until about 2 billion years ago, when we have evidence in the fossil record of what appeared to be eukaryotic cells. But we, according to our own theories, it seems that to us that eukaryotic cells appeared much later than that. But this is another discussion. And the presentation of the problem. So the prokaryotes of a prototype of that is the bacterial cell. It's basically a structure which is in some way complex, but less complex than the eukaryotic cell, and which is a structure bounded by plasma membrane, a capsule, a cell wall, and the cytoplasm, and in the center we have the genetic material, which is not bounded, which is not circumscribed by a nucleus. And in the absence of the nucleus is the major difference between a prokaryote and a eukaryotic cell. And so there are no nuclei in the prokaryotic cell. There are no membrane-bound organelles such as mitochondria or chloroplasts. And here we have the genetic material. The DNA is in the region of the cytoplasm that they call nucleoid. And later in the history of life, we have the appearance of the cells, which are much larger, several magnitude larger than a prokaryote cell, than a bacteria, much larger, and they have the nucleus. Here we have a nucleus which contain the genetic material, and other membrane organelles such as the mitochondria. In a plant cell or in the algae cell, we have the chloroplast. We have those nanostructures which are the endoplasmic reticulum. The rough one were synthesis of proteins are made, particularly in the ribosomes. We have ribosomes in bacteria, but we don't have endoplasmic reticulum. We have the Golgi apparatus, we have the microtubules, which are new structures in the eukaryotic cells, and those cells appear all of a sudden. And the question is, how did that happen? Is it Darwinian evolution? Is it by the agency of intelligent designer? So what's the explanation for the appearance of the cells, and without which you wouldn't be there, macroscopic life wouldn't be there, the plants, the animals, that a major part of our ecosystem wouldn't exist without the eukaryotic cells. So it is a question then that should interest not just scientists or professional of science, but should interest every human being, because that's a question um, that should interest all of us, that tells about our origin, and it tells us our fate and our destiny as well. Um, um, it should interest all of us. And uh, late in the last century, so we have the microbiologist Carl Woese, who came up with the organization of life. So before him, it was life was practically cell-based life was divided between prokaryotes, which were mostly represented by bacteria, and eukaryotes, which is mostly macroscopic life, human, animal, plants, and fungi, and some protists, and the protist. And but he came in, I think, around in the 1970s. He came with according to his research, he was able to decipher the presence of archaea, which is another unicellular organism in the broad group that was bacteria. So he were able to come up with a third domain of life, archaea, along with bacteria and eukarya, to which we belong. And according to his new organization, all those domains would have sprung up from a common ancestor. And here, just to give you a clear idea, some examples of bacteria, such as E. coli. That's a common bacterium that we know. That's part of that we can find in our digestive tract, but some strains of it can cause illness in the GI tract. But if the E. coli is in a different spot in the organism, as if it's in the urinary tract, you have a urinary infection. Strep pneumonia, which causes pneumonia, Salmonella typhi, which is a bacterium that causes typhoid fever, particularly in developing countries. Strep group A, which causes throat strep infection. Chlamydia trachomatis, which is a bacterium that causes sexually transmitted infection. Lactobacillus acidophilus, which is a bacterium which is normal, that's normal in our gut, part of our microbiome. And some classes of archaea, those are prokaryotes that can be found in our digestive tract as well, part of a microbiome. They don't cause illness in humans. They don't cause illness in eukaryotes, actually, the archaea. But if there's an imbalance of them, you may have the imbalance may cause diseases. But archaea are mostly called extremophile. They are found in extremes of temperature, in extremes of salinity, and or pH. Now they are called extremophiles for that reason. And eukarya, which are plants, the animals, the fungi, the protists, as we talked about before. Those are the three domains of life according to Carl. So, and so the difference between for a good part of the history of life, we had the prokaryotic cells, such as the bacteria and archaea, and all of a sudden we have the eukaryotic cell. This is the nature of the problem. So how did we go from the left to the right? What happened uh, in the middle? And here, to make a difference between the plant cell, which is an eukaryotic cell as well, and they have the the structure that we call chloroplasts, where photosynthesis is done, and the transformation of using solar energy transform in CO2 and water can form glucose from energy for that. So this is photosynthesis in action. So they have the structure that we don't find in an animal cell. All right, so this is the domain of life, and it has not always been like that. So what happened? How did the eukaryotic cell happen? Again, we have for a very long time in the history of life, there were no eukaryotes, and they appeared. Um, so the current theory to explain the appearance of eukaryotes is the endosymbiotic theory of the late Professor Lynn Margulis, which is a theory that has been initially propounded at the beginning of the 20th century by a Russian scientist. I think his name was Chosky. But well, the credit goes to Professor Lynn Margulis because she was the one to have explained and provide evidence for this theory, and really developed the theory of endosymbiotic theory, according to which the structure that differentiate eukaryotes from prokaryotes, she came up with a theory to explain the presence of the mitochondria, the presence of the chloroplasts, and of the nucleus. This is a widely accepted theory to explain the arrival of eukaryotes, and it's a busy slide, but I'll break it down to explain the endosymbiotic theory. The first part of it is that like a pre-eukaryotic cell or proto-eukaryotic cell would invaginate, would cause some enfolding of the membrane, would cause the nucleus. The first is the formation of the nucleus. It's a multi-step process to explain the origin of the eukaryotic cell. The first cell, a certain prokaryotic proto-eukaryote. Some would say it was a bacterium, some would say it was an archaea, but we're saying right now it could not have been an archaea because the archaea structure, their membranes are completely different than the membranes of eukaryotes. So our membrane for eukaryotes for very much similar to the membrane of a bacteria, but there are differential. There are some major differences, such as for instance, in the phospholipids of the archaea that are completely different than the phospholipids of eukaryotes. So there is no way that proto eukaryotic cells could have been an archaea. So we are putting this exception down completely, that the origin of the cell at the origin of the eukaryotic cell could not have been an archaea. It had to be a bacteria. So in the theory, we'll go back to the endosymbiotic theory of Professor Lynn Margulis. So invagination of the membrane and folding of the membranes will form the nucleus, and it will form also the endoplasmic reticulum. So we have the cell now from a non-nucleated cell to a nucleated cell. And then the second step would be the primary endosymbiosis, where that new nucleated cell would have engulfed an anaerobic bacterium, which is basically an alpha-proteobacteria, because it's establishing the fact that the cells from which mitochondria came alpha-proteobacteria. They said they would have engulfed alpha anaerobic bacterium, and this anaerobic bacterium would become the mitochondria that we have now. So now, from a proto-eukaryote, we have a nuclear cell, we have a nucleated cell, and now cells with mitochondria that would form the first eukaryotic cells, and such as animal cells. And the next step would be a subset of those new cells that have mitochondria would have engulfed a cyanobacterium, a photosynthetic bacterium, and then those would be the origin of the chloroplasts. So those form on top of the left here, the modern photosynthetic eukaryotes, such as an algae a cell or a plant cell. So this is the theory of this is how the theory of endosymbiotic is proposed. First, formation of the nucleus. Second, primary endosymbiosis, so formation of we have engulfing of the aerobic bacterium to form the mitochondria, engulfing of the cyanobacterium to form the chloroplasts, and then you have eukaryotic cells from that. So the endosymbiotic theory, as proposed by the Professor Lynn Margulis, it's very complex. It's a multi-step process to go from bacteria to go from prokaryotes to eukaryotes. It's a very complex development, and we are coming up with, we are proposing our book, the bilateral descent theory, which is part of the quantum theory of origins to explain the origin of eukaryotes. And in part of our quantum theory of origins, we explain three phenomena, which is the origin of life, the origin of eukaryotes, and the origin of the universe by what we call quantum origins, which is basically all of those systems started with their most simple units or most simple expressions. In the case of an eukaryote, if you take a human, if you take off all what you have as far as structure, if you start with, take your head out, take your the bones, the muscle, and if you go back to your life, if you go back to your development, and you go back, the first cell that made you is the zygote. So which is what we present here. The first cell of an eukaryote is the zygote or a seed. If it's a plant, it's a seed. If it's an animal, it's a zygote. And this is our most simple expression. And how was that formed? How was that zygote formed? Zygote or seed, the first eukaryotic, it was formed, if we take the case of human, it was formed by a sperm meeting and fusing with an egg, and then from that encounter, the fertilization of the egg, we get a zygote, which is the first phenotypical expression of a human being. Or if you take a mouse, if you take a polar bear, if you take a lion, it's the same process. When we compare that process to what happened in the microbial world, and when a bacteriophage infects a bacteria for the bacteriophage to multiply itself, it's the same process. It's a similar process of a sperm meeting an egg. And in that case, for a bacteriophage, for a virus infecting a bacterium, the virus here in the first picture infect the bacterium and multiplies inside the bacterium, and multiple variants are released, and the bacterial cell is dead. So what we're proposing is that the eukaryotic cell appeared not according to endosymbiotic theory a la Lynn Margulis, but according to this proposition that it was from the fusion of a virus, which was a retrophage, a retrovirus for bacteria, a retrophage, fusing or fertilizing a bacterium. Basically, in our theory, if you read our book, we'll see that more plainly. It was a gammaproteobacterium in the case of animals and plants as well. And the fusion of a retrophage virus with a gammaproteum formed a new cell, a cell which is a eukaryotic cell. And it was zygote in the case of multicellular eukaryotes. It was the first cell of unicellular eukaryotes, such as an amoeba, for instance. and all those zygote would lead to the different species. We know that that's going to be a new topic to explain the origin of species, but those zygotes which are formed by the fusion of a virus with a bacterium um, are responsible, are the first eukaryotic cells. Um, the bacteria involved are different for human, for animals, and for plants. And the bacteria, we found that responsible, forming the first egg of animals is a bacterium that was discovered in the late 1900, I think, 1990, somewhat by a team from the Max Planck Institute of Microbiology. It's a Tumorgarita namibensis. It's a bacterium that was found close to in the coast of Namibia. It has a lot of sulfur deposits in it. So it's a gammaproteobacterium. It's a very large bacterium that's visible to the naked eye. And again, in the quick class of gammaproteobacteria, we have some different bacteria that form at the region of the ovaries of plants, Bilateral, which is a filamentous bacteria, and also bacteria. Those bacteria give rise to algae and plants. So how that forms? So how does a virus infect a bacterium cause this bacterium to be transformed into a eukaryotic cell? So we have to explain that by that process only. It's a single step process, which is different than a multi-step process. Anyway, it's not necessarily a single step process. By that process only, we have appearance of a eukaryotic cell. First of all, there are evidence. We're going to here talk about two studies that viruses that infect bacteria, sometimes they in that process of infecting the bacterium to release new virus, they form a temporary nucleus inside this bacterium. Here we're talking about a study by Vorapunchiki and collaborators. Talking about, you can check that study as well, assembly of a nucleus-like structure during viral replication in bacteria. So they report the virus infect the bacterium, and in that process, prior to release of new virions, there's the virus form a nucleus-like structure, a compartment inside the bacterium. Particularly, it was they studied the Pseudomonas phage, which showed that it's assembled a compartment that separated the viral DNA from the cytoplasm. Here, this is a tomogram of this process. Here on the left in A, we can see here that nucleus-like compartment that's formed, while you can see here the capsid of the virus are being formed on the side here. The dark capsids are when the capsids are already filled with the viral DNA before they're being released. And on the right, you see more. It's a colored picture of that. You see the nucleus-like structure that's formed by the virus inside the bacteria, and then the capsid and also the tail of the viruses that has to be attached to the capsid before they are being released. So like this Pseudomonas phage infection of Pseudomonas chloranthus, and then it's formed a temporary. So the viruses, what we're getting at, have the capacity to form the nucleus. Another study by Mendoza and collaborators, which showed that again, bacteriophage infecting a bacterium, and to shield the DNA, the nucleic acid of the virus from the immune system of the bacterium, particularly the CRISPR-Cas system. Here on the left side, we have the cascade Cas3, Cas9, Cas12, which are basically part of the immune system of the bacterium to really cleave those foreign DNA from the viruses. It forms, so the virus, what it does, it forms a compartment, a nucleus-like compartment within the bacterium to shield the DNA of the that came from the bacteriophage form those nucleuses. And then here we have the reproduction of the DNA of the virus, feed them into those capsid heads, and then release them. Again, demonstration of a formation of a nucleus structure by viruses infecting bacteria. So to our point that a virus infecting a bacterium cause initially that bacterium to become a nucleated cell. So this is the study we just talked about, and formation of a nucleus-like compartment by this Pseudomonas phage they call ficus. And what about the origin of the other organelles, such as the bacteria, such as the mitochondria and chloroplasts? We're not going to spend a lot of time on that, but we will say that we propose in our new theory that those organelles were formed by a transduction of bacterial DNA from one bacterium to another bacterium. So some viruses, they are polyvalent, they can infect more than one bacterium. And what we propose is that the viruses at the origin of eukaryotic cells, they would have infected, for instance, an alpha-proteobacterium and get the DNA of that bacterium and transduce it into gammaproteobacterium to make this bacterium becoming a eukaryotic cell. At the same time, too, it's the same process, infected the cyanobacterium and transduce it into an ancestor of a plant cell or algae cell. And those, when they infect these first cells, the DNA material that's transduced could be becoming part of the chromosome, or it can be transduced as well as a plasmid, which is basically what almost like a mitochondria or a chloroplast look like a plasmid of a bacterial cell. But it's possible that they have transduced those DNA both into the chromosome of the cell, also as plasmid as well. So again, by one infection, the virus would be responsible for making the nucleus, making the mitochondria, not by phagocytosis not by endosymbiosis or phagocytosis of alpha-proteobacteria or phagocytosis of a cyanobacterium, but by transduction of genetic material from those previous bacteria that the virus would have infected and destroyed, and they would come up with those piece of DNA they've taken for the DNA they've taken from those bacteria and transduce them into the new bacterium that they infect, the gammaproteobacteria that will become the eukaryotic cells. So transduction, it's a well-known mechanism that has been discovered by two major scientists. I don't have their name right in there, and so I don't have their name right in my head too. But it's a well-known method of bacteria getting new genetic material. Here we have a quickly going over possible transaction. We see here a virus in number 1 with genetic material in red and the bacterium with genetic material in blue, and would infect the bacterium in 2 here. It would take a piece of the genetic material. We see here the virus on the left in number 3 has a piece of blue genetic material. It would leave that bacterium and infect another bacterium, and then would bring that donor DNA into that new bacterium, which is a receiver, would receive that DNA from another bacterium and would place it into the genetic material of that new bacterium. So a piece of genetic material from a bacterium get into a new bacterium by by transduction, viral transduction with a phage. So what are the evidence of this? That's why we call it bilateral descent theory. So the eukaryotic cell is come with from the meeting of two different ancestors, a bacterium and a virus. And we think that that new proposition for the origin of eukaryotic cell also explains the origin of sex. It's been a big unsolved question in biology about what's the origin of sex in eukaryotes. Why all the eukaryotes have sex, or pretty much most of them. So the sex in the origin of eukaryotes stem from the origin, which is from the fabric of the eukaryotes, which is basically they come from the fertilization of a bacterium by viruses, by a virus. And so we're going to talk quickly about the viral evidence, the bacterial evidence, the fossil evidence for this new theory of eukaryogenesis. And for viral evidence, so why we think that virus is important has been a key factor in the origin of eukaryotes. If you go with the Human Genome Project, one of the surprises that was found is that most of eukaryotic genomes, practically half in human, derive from retrovirus elements called retrotransposons. So half of our DNA is viral. So some scientists explain that fact, saying that in the history of humans, in the history of during the course of our history and our ancestors, we've been infected by retroviruses. Like if you take the HIV virus now, for some people that are infected, the HIV virus is able to integrate some of its DNA into human DNA. So that's a proven fact. And the thing that and then we get those viral elements in our genome from several retroviral infection. What I'm saying differently is that that happened as well, but the reason why we have so much viral or retroviral DNA in our genome is because our fabric as eukaryotes started with the fusion of a virus with a bacterial cell. So our ancestor cell is both from a virus and a bacterium. So the virus was our great grandfather and the bacterium was our great grandmother. And here are components of the human genome. All those elements here: intron 26%, line is 20%, sign is 13%, LTR, all those are viral elements, which is more than 60% of our genome is mostly viral elements. If you make the addition, again, and also even our in developmental biology, what explains our anterior-posterior axis, why we have a head and feet, why animals have a head and tail. The anterior-posterior axis, that patterning of animals, is explained by our original virus. We obtain that from viruses, particularly the codovirals, which is the virus that made us. It's basically about the same origin as the codovirals. So it's basically one codoviral that really made us. And then and we'll see here the sperms of those different mammals. They have like a head tail pattern, head tail axis, which is what's responsible for anterior posterior patterning as animals. All animals have anterior posterior patterning. Even medusae, even some other mammal marine animals have anterior posterior patterning. We'll talk about that later. And the oxygen as well in the anterior posterior patterning of animals. So we can make comparison with retroviral genes and Gagpol, the organization of those retroviral genes as well. And even some of the proteins that are very important in mammals, particularly the syncytin protein, not just in humans but in all placental animals. It's involved in the formation of the placenta, has been made from the enveloped gene of a human endogenic retroviruses. So our fabric, even of mammals, is made by by some protein that have been from viruses. So those viruses, what I'm saying, are not viruses that in the course of evolution happen to have infected our ancestors. We are made by our ancestors cells of eukaryotic cells resulted from the fusion of a virus with a bacterium. Bacterial evidence, similarities of will show for bacterial evidence quickly. Will show that the bacterium we talked about, the gammaproteobacteria that we talked about, they have a lot of similarities with the eggs of mammals or eggs of like egg-laying animals. Tumor which is the cell, which is the bacterial cell, which is the ancestor, which is the grandmother of us of all animals. So I'm saying here, one bacterium is the difference in origin of all animals are mostly the viral strains that made us. But we all, whether you take a dog, a cat, a human, a bear, a shark, and all of them will come from this bacteria, Tumorgarida nabibensis, which is the great grandmother of us all. And here, a comparison of TN Tumorgarida nabibensis bacterium compared to like a regular bacterium, which is about at least one order of magnitude above a normal bacterium. A normal bacterium could be 1 or 2 order of magnitude above, would be 1 or 2 10 microns, and so the Tumorgarida nabibensis, like mammalian egg or egg of egg-laying animal, they are they are really large structure that visible to the naked eye. Spherical, they are non-mortal, same characteristic descriptively. And for the metabolic profile of T. nabibensis, so those are bacteria that are found in low oxygen environments. They use sulfur for energy, that which they oxidize with nitrate. Nitrate mostly, that's what they use, but they can use oxygen facultatively. And if you put them in culture, they produce ammonium by the reduction of the nitrates. So this is the characteristics of the TN bacteria. They low environmental oxygen, they facultatively aerobic, they mostly use sulfur, which and nitrate as electron acceptors, and they produce in their culture media they produce ammonium. And then if you take a mammalian cell, a mammalian egg, they are found in the oviduct in low oxygen concentration, and such as the egg of egg-laying animal, they developed in oxygen deprived environment. And mammalian oocytes, a mammalian egg, the egg of any mammal, there's high production of nitric oxide and hydrogen sulfur. So they use those elements, nitrite and sulfur again. And then in the case of a egg-laying animal, those animals use a lot of they accumulate nitrogen, they accumulate sulfur-rich amino acids, like taurine supplementation in their diet for some egg from egg-laying animals. It's a limiting factor for egg production. So dietary taurine supplementation promote egg production. So again, same thing for the Tumorgarida nabibensis bacteria. They use nitrate and sulfur. And then if you take in the culture media, like say in IVF culture, you find that mouse and human embryos, they generate a lot of ammonium in culture media. Same thing as also as eggs of oviparous animals. Same thing as Tumorgarida nabibensis, they produce ammonium, which make that relationship between eggs of animals, and Tumorgarida nabibensis bacteria. They have a lot of metabolic similarities. The fossil evidence I will talk about is area in southern China. they call the Dushan Formation in southern China, where they found a lot of egg fossils, embryo-like fossils that mostly in the Precambrian. So that's what they found microscopically. A lot of fossils, and without finding any animals, they found those egg fossils like 2-cell division, 3-cell division, and more cell division that in in the fossil record there. And here, which make uh, for the history of life, which is a graph for the history of life. At the bottom, we have those in the Precambrian, late Precambrian, we have those eggs and then those early embryos without finding any animals. And later on, you have in the late Precambrian, you have the formation of those um, of the structures of the Precambrian animals, which were like soft-bodied animals. And later in the Cambrian, you have the trilobites, who have all this Cambrian explosion of fossils animals. So from eggs to soft-body to hard shell animals in the Cambrian. So which again support our quantum theory of origins, that eukaryotes started with first the eggs of eukaryotes, I mean the seeds or the zygotes, and then we have kind of a development of an embryo, and we have um, soft-bodied animals, and then you have hard shell animals later on. In summary, in lieu to explain the origin of eukaryotes, in lieu of the endosymbiotic theory of Lynn Margulis, we're coming with a new theory to explain eukaryogenesis, which is the bilateral descent theory, which is eukaryotes came from two different structures: a virus and a bacteria that had fused together to form a new cell, a seed for plant, a zygote for animals, like a unicellular structure for unicellular eukaryotes. So the fusion of a virus with bacteria made a new zygote or unicellular. So this is how we're proposing that for the origin of eukaryotes. And thank you. We would appreciate your comments and questions. We'll try to answer as much as we can. But now, see you for the next video. Thank you.